Good morning, all of you. Today, we'll be discussing the most probable questions that are likely to be asked for this year Neet examinations from the chapter 18, that is body fluids and circulation. Okay? So, the type of questions as well as the content of the questions you have to take into consideration. We expect the same type of questions and the same material content to be asked this year. Okay? We are all going to the questions directly. The first question on the board. Give the correct option that indicates donors group for the blood group A, B, ABO respectively. See, in blood grouping, we will be having a donor and a recipient. A donor and a recipient. One who donates the blood, that blood is having antigen and antibody. One who receives the blood, recipient blood, that is also having antigen and antibody, and reactions can occur with the antigen of the donor with the antibody of the recipient. That is more important clinically. So, antigen of the donor reacting with antibody of the recipient, that is very, very important. So, donor's blood group for A. So, who can all donate? The antigen. Remember that? In receiving blood, the recipient's antibody is to be given into account. For example, A group is containing anti-B. He is having A antigen. Okay. And he is having anti-B. So, he cannot receive blood that will be containing an antigen that reacts with the antibody, that is anti-B in his blood. That is the clue. That means he can receive blood from what group? A group. Because A group is having A antigen and he is having antibody B. So there is no problem. He cannot receive blood from B or AB because they are having antigen B and he is having antibody B. So he cannot receive blood from B or AB, but can receive blood from O group because O group is having no antigen. So only A and O he can receive. For the same reason, a B group person is having antigen B and antibody A. So, he cannot receive blood from A group because A group man is having antigen A and he is having antibody B. They will be reacting. So, he can receive blood from B group and O group. And AB group. See, AB group is having both antigen but having no antibody. Since he is having no antibody, he can receive any blood, any group from any person. That's why AB is usually called universal recipient, because he is having no antibody at all. And O group, he is having no antigen but having both antibody. Since he is having both antibodies, anti-A and anti-B, he can receive blood from a person who is having no antigen at all. So, who is having no antigen at all? O group. He can receive blood only from O group. So, that is the correct answer. Is that point clear? All of these are wrong. The clue is simply this one. Donor's antigen reacting with the recipient antibody. wrong and correct statements. Plasma contains 55% of human blood. See, plasma, which is abundant, constitute 55% of the human blood. Albumin, corpuscles are 45% and plasma is only 55%. We'll wait. Albumin is a plasma protein which helps in osmotic balance. Correct? Albumin is responsible for keeping up water with the body. Factor responsible for the clotting process are present in the blood. Yes, that also is a correct one. Plasma constitutes, factors responsible for the clotting process are present in the blood. Blood contains clotting factors. Plasma without clotting factor is called serum. When the clotting factors are all removed, we call it as serum. Minerals are not generally found in blood. Minerals are present and they constitute 1 to 2%, mainly in the form of chlorides, phosphates, carbonates, etc. of sodium, potassium, calcium, magnesium are present. They constitute about 1 to 2%. So, the answer is only 5 is wrong. The marking is wrong. Only 5 is wrong and all other 1 to 2, 1 to 4 are correct. This is the correct answer. Okay? What is marked here is a wrong one. Identify the correct sequence of cardiac cycle. During a cardiac cycle, what are all happening? Of this, here only 4 are given. Remember, atrium contracts first, followed by ventricular contraction. They are not contracting simultaneously, but one after another. So, atrial systole contraction, that will be pumping about 30% of the blood to the ventricle. So, ventricle become full with blood. It is called ventricular filling. Then the ventricle will be contracting. Then the ventricle will be relaxing. Okay. During the ventricular systole, the atrium will be relaxing. They happen simultaneously, coinciding with each other. Now, after ventricular diastole, both atrium and ventricle will be in the same stage, diastole. This is called joy and diastole. The cardiac cycle starts from a stage called joy and diastole. What is that joy and diastole? When atrium and ventricle both are in a fully relaxed condition. Clear? In a cardiac cycle, duration of different events are presented. Remember, in 1 minute, average 72 times the heart is undergoing rhythmic beating. That is, the time taken for one cardiac cycle will be 60 by 72, 60 seconds in 1 minute. 72, that is 0.8 second. That is the time for one cardiac cycle. Okay, for one cardiac cycle, the time taken is 0.8 second. And identify ABC respectively. 0.1 second is for the atrial systole. So, A is atrial systole. B, 0.3 seconds, ventricular systole. And what is C? During which both atrium and ventricle together are in resting state, diastole. That's called joy and diastole. Okay. So, we have identified them. Then, duration of B and C put together. B and C, 0.7 second. See, for 0.1 second, atrium is systole. Atrium is contracting for 0.1. That means for the remaining 0.7 second, it is in diastole. So, atrial diastole, 0.7 seconds. Duration of C and A together, 0.4 and see, this is during this phase that ventricle is in systole. That means in other time, up to this, the ventricle is in diastole. So, ventricular diastole, this is 0.4 and 0.1, so 0.5 seconds. So, ventricle alone in diastole for 5 seconds. Out of that 5 seconds, for 0.4 seconds, atrium and ventricle together. See, they are all remaining in a joint diastole for 0.4 seconds. And when atrium is in systole, that time also ventricle is in diastole. So, this time plus this time, ventricle is in diastole. So, out of the 0.5 seconds, for 0.4 seconds, it is together with the atria in diastole. Clear? That's why we go to the answer 1. Atrial systole, ventricular systole, joint diastole. Atrial diastole, ventricular diastole. Correct statement. Complete double circulation is present in all tetrapods. Tetrapods include those which are having 2 pairs of limbs, amphibia to mammals, and only in birds and mammals you are having complete double circulation. Why? Because alone in them we are having complete separation of the ventricles. Okay, so all tetrapods they are having complete double circulation is wrong. In amphibia and reptilia we are having incomplete double circulation, whereas birds and mammals alone we are having complete double circulation. Myogenic heart. What is myogenic heart? Heart whose activity is initiated and maintained by the muscles of the heart itself. The rhythm is maintained by, initiated and maintained by the heart itself. That's called myogenic. Vertebrate heart is myogenic. Molluscan heart is also myogenic. Okay, so the myogenic heart is a feature of molluscan vertebrates. Correct statement. Thickest muscular wall in human heart is applicable for left ventricle. Left ventricle is the largest chamber whose walls are is also the thickest. Correct. In vertebrates, organ which receive deoxygenated blood is lungs. See, we are all supplying venous blood to the lungs for oxygenation because lungs are the organs where exchange of gas are taking place. So, blood coming to the heart, coming to the lungs will be deoxygenated and going out will be oxygenated. That's a correct statement. So, we are having one only incorrect, 2, 3 and 4 are the correct ones. Okay. Which of these are inactive forms? In blood clotting, a total of 13 factors are involved and some of them are present in an inactive form. Fibrin is an active one, but fibrinogen, that is how they are all present in the blood. Fibrinogen is an inactive one. Fibrin is active. Thrombin is active. Prothrombin is active. Thrombokinase is an activating enzyme that converts prothrombin to thrombin. It is an active one. Thrombokinase is also called thromboplastin. It is an active one. So, those who are all inactive are 2 and 4. Okay? 2 and 4. Of the 13 factors, you have to study at least 4 important factors. The first one, fibrinogen. Second one, prothrombin. Third one, thrombokinase or thromboplastin. And fourth one, calcium. That calcium is responsible for conversion of fibrinogen to fibrin. Without calcium, no clotting can occur. That is the principle in adding a small amount of citrate to the blood when lab people are collecting blood for dialysis or for diagnosis. So that will be absorbing all the calcium. Once calcium is removed, blood will not float at all. That is the principle. So presence of calcium is a must. Choose the correct pathway of transmission of impulses. Remember, our heart is initiated and maintained by the muscles of the heart itself. There are certain specialized tissues called nodal tissues. The most important nodal tissue is the SA node, sinoatrial node. Second node is the atrioventricular node or AV node. So signal from SA node, which is the pacemaker. Okay. This pacemaker, SA node, is having high rhythmicity, 70 to 75. Average we take 72. And its conductivity is slow. That is, the signal will be traveling at a low rate of about 0.3 meter per second. That's why we say that rhythmicity is high, but conductivity is low for SA node. But for AV node, it is also auto-excitable. Its conductivity is very high. It can conduct impulses at a rate of 1.5 meter per second. But its rhythmicity is only 45 to 50 per minute. Frequency is less, 45 to 50. Okay. So from SA node to AV node, then it passes through the bundle, AV bundle. Then after reaching the septum, it is dividing into two, anterior and posterior bundle of his. And after reaching the apex, they branch into small branches called Purkinje fibers. So SA node, AV node. Okay. From AV node, AV bundle. Then bundle of his Purkinje. That is the order. So AV node, no. SA node, this is correct. AV node, no. SA node, AV bundle, no. It will be first reaching the AV node. SA node, Purkinje, no. This is the order. Clear? Hemoglobin of human fetus. There is a small difference in the nature of hemoglobin. What is that difference? Now I am telling you. We know the oxygen dissociation curve for hemoglobin of adult, HbA. What is the peculiarity of HbA? We are having 2 alpha chains, 2 beta chains. 2 alpha chains and 2 beta chains. And for fetal hemoglobin, there is a difference. We have two alpha chains and 2 gamma chains. And this is having a more affinity with hemoglobin. This hemoglobin is having more affinity with oxygen. Fetal hemoglobin is having more affinity with oxygen. That's why the curve is more to the left. See, the P50 value, that is the partial pressure at which 50% hemoglobin becomes saturated. In our case, is 27. But for hemoglobin F, this P50 value is 19. That is, even at a partial pressure of 19, Even if the oxygen partial pressure is 19, hemoglobin of a fetus become 50% saturated. 50% saturation. Whereas we need a higher partial pressure. Only in a higher partial pressure, our hemoglobin become saturated 50%. So there's a gradual change. As a fetus is developing or adult, the percentage of fetal hemoglobin is decreasing. Even in a fully developed adult individual, we will be having 5% of our hemoglobin of HbF category, hemoglobin F. So this is having more affinity. It has a higher affinity for oxygen than that of an adult. Is that clear? Hemoglobin of fetus is having two gamma chains and no beta chains. There is no change in the number of amino acids that are present, but their position is slightly different. That's all. And there are some differences between these two also, other than the chemical nature. Adult hemoglobin will be influenced by the presence of certain chemicals in the tissues. DPG, diphosphoglycerate. This diphosphoglycerate will be acting through the beta chain of adult, causing more oxygen to be released. So dissociation of oxyhemoglobin will be favored by the presence of DPG, diphosphoglycerate. Okay, 1,2-diphosphoglycerate, and this DPG is having its affinity or reaction with the beta chain, and since there is no beta chain, this DPG will be having no action with the hemoglobin of fetus, HbA, hemoglobin of adult and hemoglobin of fetus, HbF. Okay, so DPG is having a negative influence with the HbA but having no influence with the hemoglobin of fetus, because in fetal hemoglobin there is no beta chain, diphosphoglycerate. Okay. Which is the correct sequence of arrangement of type of WBC in increasing order in terms of number. So it is based upon the number, not based upon the size. Different types of WBC. Okay, this is the diagram for your reference. So you need not worry about these ones. Eosinophils, 2 to 3%. Basophil, the least one, 0.5 to 1%. Neutrophil, 65 to 70%, most abundant. Monocytes, 6 to 8%, and lymphocytes, 20 to 25%. We know that. Now arrange them. In what order? In decreasing order. So the first one should be the most abundant one. Which is the most abundant one? Polymorpho neutrophils. So neutrophils are the first one. So you can immediately eliminate all other options. Even if you don't know the correct value of other, there is a technique of arriving at the correct answer. How? We know that polymorpho neutrophils is the most abundant one. So it is in a decreasing order. So the first one should be always the one which is having the maximum abundance. So that is for the polymorpho neutrophil. Then eosinophils and basophils. Eosinophils are 2 to 3%. Basophils are only 0.5 to 1%. Least basophils. Clear? Find the incorrect match. Chordae tendinae. What are chordae tendinae? The fibrous tendinous connections between the flaps of the bicuspid and tricuspid valve, cusp of these valves, with the papillary muscles, bundle of his, two bundles of conducting system, Purkinje fibers, right and left bundle, together known as bundle of his, correct? Purkinje fibers, minute fibers, correct? Throughout the ventricular musculature, which is branching, correct? Fossa ovalis. See, this is a term not given in our books. What is fossa ovalis? Fossa means a closed window. It is, see, during development, in the fetal condition, the impure blood that is reaching the right atria will be entering into the left atria through a small opening, a temporary aperture in the interatrial septum. This opening is foramen ovale. Foramen means opening. We are having foramen magnum in the cranium. Foramen ovale is the oval window. Fossa ovalis. See, fossa ovalis, closed window, is a remnant of foramen ovale. Foramen ovale is present in adult heart. That is wrong. So, this opening is the foramen ovale. So, the blood will be reaching the right atria from different parts of the body, and they will not be sent to the lungs for oxygenation because lungs are not functional. Lungs become functional only after delivery, only when development is complete and it has become an adult. Okay, till that time, placenta is doing the functions. So, the blood need not be sent to the lungs for oxygenation. So that from the right atria through a bypass opening present in the interatrial septum, it reaches the left atria. That opening is called foramen. Foramen means opening. It is present in fetus. Okay, and this is the aorta that takes pure blood. Blood vessels are there. And this is the pulmonary artery that is taking impure blood, but it is not functional. So, even if there is any small amount of blood that is entering from right atrium to the right ventricle and trying to pass through this one, that blood is shunted to the aorta. It is not allowed to go to the lungs because lungs are not functional through a small duct. See, you are having a tremendous change in the circulatory system as we are all developing from fetus to an adult. You are having ductus Bottali or ductus arteriosum that connect the pulmonary and the systemic. So, what are the two peculiarities present in the fetal heart? Presence of a foramen ovale, 2, a small blood vessel that is connecting the systemic with that of the pulmonary, so that any blood passing through the pulmonary is again brought back to or given to the dorsal aorta. Now, when the development is complete, this opening become closed. This opening become closed down. That closed window is called fossa ovalis. So that fossa ovalis is the remnant of the foramen ovale present in adult. And foramen ovale is present in fetal heart. Because the lungs start functioning, lungs are the oxygenating agent, so blood has to be given to the lungs. So this opening is closed, and from right atrium, blood will be reaching the right ventricle, and through the pulmonary artery, blood is reaching the lungs, and it should not pass through this one. So this blood vessel become closed, and that closed non-functional duct, ductus botalli or ductus arteriosum, is now called ligamentum arteriosum. So what are the two changes that are occurring? One, fossa ovalis, foramen become closed and become fossa ovalis. Ductus botalli or ductus arteriosum become a ligament, a connecting structure between these two. That is called ligamentum arteriosum. These are not in our books. So understand? The foramen ovale is present not in adult heart but in infants. Then ventricular systole. Ventricle is contracting. So when ventricle is contracting, what will happen? The volume of the ventricular cavity become ventricular chamber become reduced, pressure increases. Blood to flow into the ventricles. See, ventricular systole causes. When this is contracting, what happens? Actually, the blood is flowing, see, to the arteries, from the right ventricle to the pulmonary artery, and from the left one to the aorta. And these valves are closed, tricuspid and bicuspid valves are closed. And semilunar valves that guard the aorta, pulmonary artery as well as the aorta, are all open. That is a change occurring. So blood to flow into the ventricle is wrong because ventricle is contracting. Blood to flow out of the atria, no, the atrioventricular valves close. What is the atrioventricular valves? They are the the tricuspid and bicuspid valves. They are all closed and the semilunar valves are opening. So when ventricle is contracting, pressure is increasing. That increase in pressure causes the tricuspid and bicuspid valve to be closed and the semilunar valves to open. Thus, blood will be entering into the aorta and pulmonary arteries. Okay. Semilunar valves to close, no, they are all opening. Pressure to decrease, no, pressure is increasing. Clear? Then, fibrinogen. What is fibrinogen? Fibrinogen is responsible for blood clotting. So, blood coagulation. A is one. Globulins, of which immunoglobulin, that is antibodies, which are all important in the defense of the body. And albumins, which is responsible for keeping the water balance of the body. So, it is ABC 1, 2, 3. So, the option is DOL. Again, we have discussed the earlier question. You can refer that. Lymphocytes. Lymphocytes are 20 to 25%. So, A is 2. We are now concentrating only C and D because all of these are wrong. And what is their function? We are having lymphocytes. Lymphocytes are 6 to 8 in nature. Okay, that we agree. And they are smaller ones, smallest of the leukocytes. Okay, there are two types, larger and smaller. Neutrophils, most abundant, 60 to 65. Okay, we are also having the same one, but what is the function? That is the macrophagic cells, multi-lobed nucleus. That is why it is called polymorpho neutrophils, 2 to 7 lobes. That is why they are polymorphic. Some cells will be having 3, some will be having 5, like that. So polymorphic. So neutrophils, B should be 3 and 1. So immediately we came to the final answer. Anyhow, we'll continue. Basophils, C, they are the smallest among the leukocytes. Okay, we can have the smallest the basophils are the in number 0.5, least 0.5 to 1. And how you can identify? They are all having two to three lobes, almost like this. These are all granulocytes. So the nucleus will be almost like this. Okay. And eosinophils, always two-lobed. So based upon the nucleus, we can identify these types of WBCs by staining them first and then observing them. Okay. Monocytes are the largest in size. Okay. 6 to 8%, they are having a nucleus which is kidney shaped, or that is the characteristic of monocytes. 6 to 8% bean shaped nucleus or kidney shaped nucleus. It is better called as kidney shaped. Okay. Kidney shaped or bean shaped. So that is the correct option. Events in blood clotting. The blood clotting is a cascading event. A number of events are occurring simultaneously. When a wound occurs, the damaged tissues and the broken platelets are all producing thromboplastinogenase that act on thromboplastinogen. Thus, active thromboplastin or thrombokinase is produced. That thrombokinase start the events. Thrombokinase will be acting on prothrombin. Prothrombin is converted into, see, I will write the events in a very simple form. Thromboplasminogen is an inactive compound. Thromboplasminogenase is an enzyme that converts this thromboplasminogen, gen is an inactive one, to active thromboplastin. Thromboplastin is also called thrombokinase. It is an activating enzyme. This thrombokinase acts on a protein which is present in inactive form, present in blood, produced by the liver under the influence of vitamin K, prothrombin. This prothrombin, it is a pro, inactive, is converted into active thrombin in presence of calcium. Prothrombin is produced under the influence of vitamin K. So it is in the liver that prothrombin is produced. Okay. Once thrombin is produced, the thrombin will be activating the next fibrinogen, which is a gen, inactive, to active fibrin. This fibrin is in next turn polymerized to form fibrin threads. These fibrin threads are forming a meshwork, a network at the site of the wound in which formed elements are trapped. Trapped elements plus meshwork will form what is called clot. After clot, what is remaining in the plasma may be oozing for some time. That's called serum. Okay, this is the overall reaction in a nutshell. And as I told you, 13 factors are involved. These factors are fibrinogen 1, prothrombin 2, thromboplastin 3, calcium 4. Without these factors, blood will not clot. You will be studying that. Factor 8, 9, 11. Because of the deficiency of these factors, you are having hemophilia. Classical hemophilia. That is hemophilia A, then hemophilia B, hemophilia C. 8, 9 and 11 factors. Okay, so we are having a lot of such events. Now coming to the question. What is correct event of occurring? So conversion of fibrinogen to fibrin. See, fibrinogen to fibrin, it is not the first one. So thromboplastin formation among them, that is number 1. So the chance is for this one. Thromboplastin. Then formation of the clot at the end. Conversion of fibrinogen before that. Conversion of prothrombin to thrombin. See, prothrombin to thrombin. You can see it here. Prothrombin to thrombin. Okay, so that should be the second one. Then 3, fibrinogen to fibrin, and finally clot. That should be the order. So the option goes to box. Clear? So it's a long cascading reaction. What's a cascading reaction? Long metabolic chain in which each step is amplified, strengthened in reaction. That's what's called a cascading reaction. What is stroke volume? What is the amount of blood that is pumped during one stroke, during one contraction? That's called stroke volume. That's about 70 ml. So 70 ml of during one cardiac cycle. So one should be C. So we eliminated all others immediately. So the tie is between C and D. Now the difference is in the second one itself. So in the second point itself, we will be arriving at the answer. What is cardiac output? Total volume of blood that will be pumped from each ventricle in 1 minute. In one contraction, 70 ml is pumped. And the heart is contracting 72 times average. So stroke volume into frequency is cardiac output. So 70 into 72, we take the average. 5 liters or 5000 ml. So second should be D. Immediately we came to the answer. See how fast you are arriving at the answer. Even looking into all the details. SAN, sino-auricular node. This sino-auricular node is having 70 to 75 times per minute. The rhythmicity. P wave. What is P and T waves? These are all the waves in a ECG. P wave denotes depolarization of the atria. So P wave, atrial depolarization. And what is P wave? Coming back, repolarization of the ventricle. Ventricular repolarization. See, ventricular depolarization and atrial depolarization. See, there is a small correction to be made. The ventricular, it is not depolarization, repolarization. This is depolarization of the atrium. This is depolarization of the ventricle, and you are having coming back of the atria occurring simultaneously with this QRS wave, and T represents the repolarization coming back, that is atrial the ventricular diastole, depolarization of the ventricle. That should be the correct one. Pick out the features not common to erythrocytes and thrombocytes. Both of them are formed in the bone marrow, correct? All blood cells are hemopoietic stem cells are giving rise to RBC and WBC. And thrombocytes carries oxygen and carbon dioxide. No, thrombocytes are having no such role. They are having role only in blood clotting, no role in transportation of gases. Both of them are enucleated, that is correct. By mature, RBC is having no nucleus shape other than spherical. RBC is by concave disc, and thrombocytes are having no shape. There are platelets. Formation is known as hemopoiesis. Blood cell formation is generally called as hemopoiesis. So, only B can be the correct answer for them, which is not common for these two. Others are all common. Aortic semilunar valves, bicuspid valve, pulmonary semilunar valve, tricuspid valve. Now, arrange them in the order in which erythrocytes will pass. That is, blood cell will be passing through them after returning to the heart from the left arm. Whether it is coming from the left arm or right arm, don't get confused. All blood which are venous from other parts of the body are all reaching the right atria. You are having superior vena cava, right, and left superior vena cava. That is bringing blood from the arms, but they are all open to the right atria. Don't worry about that. So, from right atria, that impure blood will be reaching the right ventricle, passing through the tricuspid valve. Okay, so first you should have the tricuspid valve. Immediately we came to the answer because 4 should be the first one. There is only one option where 4 is 1. See, this is not. Now, we'll discuss. Now, it reached the right ventricle. Then it passes through the semilunar valve of the ventricle. Okay, we are having the semilunar valves. So, after tricuspid, we are having pulmonary semilunar valves. Then it reaches the lungs. After oxygenation, it is coming to the left atria, and after reaching the left atria, it is reached the left ventricle after passing through the bicuspid valve. Then from here, it is pumped to the arteries, and at the beginning of the artery, you are again having semilunar valves, aortic semilunar valves. So that is the correct answer. Is that clear? So, the blood passes these route before leaving the heart. So, one point you have to write. The blood reaches the right side of the heart first, then reaches the left side only after passing through the lungs. Now, blood from the left side again reaches the right side of the heart after passing through the systems of the body. That is the beauty. The right and left heart parts are not at all connected together. Our heart is a double pump. The right side is called the pulmonary heart and the left is called the systemic heart, double heart. Now, what is this QRS complex in ECG? This is representing the depolarization. See, muscles are in a polarized condition. Now, this causes depolarization. The depolarization causes contraction. Contraction of the ventricle. Depolarization of the ventricle is contraction of the ventricle. Depolarization, and this is the repolarization of the ventricle. This is the depolarization of atria. Clear? These are represented as P, QRS and T waves respectively. Eosinophils. See, that diagram is a misplaced one, no problem. Eosinophils are concerned with allergy. If you are having eosinophilia, you will be having runny nose, etc., etc. And that is because of the eosinophils that you are having release of histamines, destructive enzymes. So it is A3. And immediately you came to the answer. You eliminated all of this. Okay, we will pass through. Even if you don't know this answer, let us see. Basophils, that is responsible for the release of granules containing histamines. So, histamine, serotonin, etc. are released by the basophil. That is given in our book. So, B is for, and again, only one option, again it is confirmed. Then neutrophils. Neutrophils are macrophagic amoeboid cells which are all involved in phagocytosis. See, the most important macrophagic cells of the blood are polymorphoid neutrophils and monocytes. And last, lymphocytes. Lymphocytes are responsible. There are two types, T cells and B cells, responsible for our immunity. So the answer is clearly the third one. Two separate circular pathway, as I told you, which is having ventricles completely separated only in birds and mammals as a whole. In reptiles, only crocodiles having, not all reptiles, only crocodiles. So we cannot take crocodile as a whole having a double complete circulation. Okay. Crocodiles are having complete double or completely separated ventricle, but they don't have still a complete double circulation because the pulmonary artery and aorta are connected by a small opening called foramen of Panizza. Because of that, some degree of mixing of blood is taking place. So, in reptiles, incomplete double circulation, even in crocodiles. So, we are having shark. Shark is a fish having single circulation. Frog, incomplete double circulation. Lizard, reptile, incomplete double circulation. Whale, a mammal. See, there is no mixing. The blood is always flowing in these two paths. You are having two circulations. One circulation to the lung, lesser circulation or pulmonary circulation. Second circulation, greater circulation or systemic circulation. Blood is never mixing together. This is because that ventricle is completely separated. Doctors use the stethoscope to hear the sound produced during each cardiac cycle. They are the left sound and the dub sound. The first sound is the leb. It is slow pitch. Longer sound lasts for 0.16 to 0.19 seconds. Okay. It is caused because of the ventricular contraction on the beginning of the ventricular systole. So, ventricular systole that increases the pressure so that tricuspid and bicuspid valves close with a thud. That is the reason for this sound, left sound. Whereas the dub sound, it is a high pitch, 0.1 second, short duration. Okay, and it is caused because of the diastole, ventricular diastole, beginning of the ventricular diastole. When diastole begins, the pressure inside falls. Pressure inside the ventricle falls. It was the high pressure that caused the semilunar valves to open. Now that increased pressure is decreasing. Now the valve is closed. Semilunar valves, they are all closed. That is the reason for the second sound. So what is the first sound? Lep sound. What is the duration between the lep and the dub? It is 0.3 second. What is the duration between the dub and the lep of the next cycle? 0.5 second. Okay, so the second sound is dub, caused because of AV node, no, AV valves, no, ventricular valves vibrate, no, semilunar valves closed, semilunar valves closed after the blood flow into the vessel from ventricle. See, this is a confusing one. After the blood flows into the vessels from the ventricle blood flows into the pulmonary artery and aorta when it is contracting. After that, so after systole, that's the meaning. After systole, it is diastoling. So during that, ventricular diastole, that's the meaning of that word. Is that clear? Name the blood cell whose reduction in number can cause clotting disorder. This is what's happening in dengue. Hemorrhagic dengue, when the platelets are falling below 60,000 per ml, it will be causing severe internal bleeding. Okay, so thrombocytes. Thrombocytes are, see, when the cells are above normal height, we call as cytosis. Erythrocytosis, leukocytosis, thrombocytosis, and low level is called penia. Erythropenia, leukopenia, thrombopenia. So it is thrombopenia, low level of thrombocytes, thrombopenia. Is that clear? See, what is serum in the clotting question I have told you. After clotting, are the clotting elements from the plasma removed? What is remaining is a colorless liquid that will not clot, because from that all the clotting elements are removed. That's called the serum. Plasma without clotting factors, lacking clotting factors. Serum differ from blood that lack clotting factors. Clear? Very simple one. Now, what is lymph? It is plasma without proteins but with some WBCs. Plasma without proteins. Large size proteins will not come, but some WBCs will come. It does not contain RBC, thrombocytes, etc. Okay, so adult human RBC are enucleated. Which of the following statement is most appropriate explanation for this feature? See, during the RBC formation, we are having erythroblast, large nucleated cells, biconvex. They are to be converted into biconcave disc, and removal of the nucleus is occurring at that time. That's called maturation. And it is for this maturation that vitamin B12, cyanocobalamin, is essential. Okay, so they do not need to reproduce. There is no nucleus. It is biconcave. So that biconcave shape is having some advantage. What is that? It is having 20 to 30% more surface area than this one. So that exchange of gases can take place through the surface, because surface area is increased to 20 to 30%. And since there is no nucleus, the entire interior is filled with hemoglobin so that it can carry maximum amount of oxygen. Okay, so they do not need to reproduce now. There are somatic cells. They do not metabolize. All their internal space is available for oxygen transport. That's the correct one. They are having two advantages also. The surface is increased to, surface area is increased to 20 to 30%. And without nucleus, all the entire, there is no mitochondria, no such structures are present. So the whole interior can be filled with hemoglobin. So maximum amount of oxygen can be transported. Clear? This is called maturation. Erythroblast convert into erythrocytes. For that, vitamin B12 is essential, that we have discussed in one of the earlier chapters. See, frog egg, when taken out of the body, continues to beat for some time. See, we are having always news. Human heart is taken from one city, it is taken by either by road or even air ambulances to another city and transplanted to another man. How is it possible? The heart goes on continuing even after removal from the body. Why? Because our heart is auto-excitable, it is myogenic, its contraction and dilation are all initiated and maintained by the heart itself. It is autogenic for vertebrates. So, frog is only a typical one, just to confuse you. It may be a frog, man, or any mammal or any other vertebrate. Frog is poikilothermic, no. Frog does not have any coronary circulation, no. Without coronary circulation, how the walls of the heart are having oxygen received? Heart is myogenic in nature. It is auto-excitable, self-excitatory. See, a neuron is not self-excitatory. There must be a stimulus, then only a neuron become excited, active. Whereas this AV nodes are all self-excitatory. Even without an external stimulus, they will go on active rhythmically. Okay, so 3 and 4. Okay, 3 and 4. Again, we are having the same. Where is the tricuspid valve? Here you are having the tricuspid valve between the right atria and right ventricle. You are having the tricuspid valve. Between the left atria and the left ventricle, you are having the bicuspid valve. And semilunar valves are at the beginning of the, see here, beginning of aorta and pulmonary arteries. So tricuspid valve between right atrium and right ventricle, bicuspid between this one, and semilunar valves are between the right ventricle and the pulmonary artery. So semilunar valves are present at the beginning of these arteries. Column 1 and column 2, fibrinogen. These are now very simple questions because we have discussed the same question earlier. See, these are all questions that were all asked in the NEET examinations. We have collected all the questions that are all asked from this chapter for the previous NEET examinations and have presented before you. So, fibrinogen. Fibrinogen is responsible for blood clotting. Globulin, immunoglobulin, responsible for the protection of the body, defense mechanism, and albumin for osmotic balance. Same type of question we have discussed earlier and is very often repeatedly asked. Again, here you are having. What is P wave? Depolarization of atria. Depolarization of atria. QRS wave. Depolarization of ventricle. T wave, repolarization of ventricle. Okay, so P wave, depolarization of ventricle, no, depolarization of atria. So it should be 4. And you eliminated other options. Only 1 and 4 you can select. Now, QRS complex, that is depolarization of ventricles. So one. So now we have come to the last one. T wave. What is T wave? Repolarization of ventricle. Repolarization of ventricle 2. And reduction in the size of the T wave. See, what is T wave? T wave is representing the repolarization, coming back, dilation of the ventricle. That is reduced means there is some problem. It is not effectively pumping. Pumping is reduced. Heart is suffering from some disease. What disease? What is the disease here? You are having coronary ischemia. What is the meaning of ischemia? Pain. Heart pain. Heart is suffering from some disease. It may be because of the crack of the muscles. It may be a heart attack. Okay. So coronary ischemia means painful heart functioning. Heart is not functioning properly. That's why you are having a change in the T wave. So the answer only we can arrive at this end. Okay. So reduction in the size of the T wave. This frequency or change is PQ interval. This is called PQ interval. PQ interval may be changed. It may be increased because of an inflammation on the atrial wall or inflammation in the AV node. Or it may be the man may be suffering from heart attack or rheumatoid fever. There may be so many reasons why there may be an increase in PQ interval. But T wave, it is not of the normal size, means it is not coming back. It is not dilating. It is coming back or dilating back to the normal size. He is suffering from coronary ischemia. Is that point clear? And coming to the last question of this paper, what would be the heart rate if a person, see, there will be always one simple question that will make you confused. The question is put in such a way that you are made to confuse. Just like in respiration, I have told you, a person, if the cardiac output is 5 liter, what is cardiac output? It is stroke volume into frequency. Here, cardiac output is, what is the amount of blood pumped in 1 minute from one ventricle? This is 5 liter. That's almost normal blood volume. If the ventricle at the end of the diastole is 100 ml and at the end of the ventricular systole is 50 ml. See, at the end of the diastole, that is when fully dilated, it is having 100 ml. Okay. And at the end of the ventricular systole, contraction, 50 ml. That means 50 ml is pumped. If 50 ml is pumped in one contraction, okay, stroke volume is 50 ml. The stroke volume is 50 ml. Okay, then what is this frequency? So, this can be obtained by 5000 divided by 50. Okay, so it is 100 beats per minute. Because cardiac output is amount of blood that is pumped from one ventricle in 1 minute. Amount of blood pumped from one ventricle in one stroke, that is stroke volume, multiplied into frequency. Okay, frequency, stroke volume. Stroke volume is 50 ml, that we calculated, and we are getting the cardiac output 5000. If for every contraction 50 ml is pumped, how much times it should contract to pump 5 liters? It should contract 100. So the heart is suffering from what is called, can anybody tell me? Tachycardia. Increased rate of contraction is called tachycardia. And the opposite is bradycardia. And with that, I come to the conclusion of this chapter discussion and wish it is highly beneficial for you. You have understood the concept, you have understood the chapter, and with these words, I conclude my session and will come before you with another set of questions next time. Okay? So thank you all. Thank you very much.